So our topic is limiting factor. Now, how do we define limiting factor? Very first thing. How do you define limiting factor? You might have learned this in chemistry as well. What is limiting? Is my, am I audible? So, what do you think? What is limiting, or what is known as limiting factor? Like the limiting reactant, you might have learned, or you must have learned it in
Yeah,
chemistry.
like something that like stops you getting like the entire like maximum capacity, I guess.
Maximum capacity, yes. So it's that factor which is actually determining your rate of reaction.
Okay.
Okay, so if it's present in a lesser amount, the reaction is going to be limited. It's not going to be like on its full extent. Okay,
Okay.
so for example, let's discuss about light. So we say why or how one factor is limiting, because the other factors are present in the maximum amount,
Okay.
or in their fully capacity, in their full capacity. For example, if we're giving something the optimum temperature that is fully available,
Okay.
if the light is still increasing, it's fully available, so definitely it's not a limiting factor. So let me make a graph for like light intensity, let's say. So we say, if we make a graph, where is my line going? Let me correct the line first. Okay, again one more line. I don't know why I'm not able to select this. Okay, so here we have a rate of reaction or rate of photosynthesis as we are discussing,
Hmm.
and on the x-axis we have light intensity. So the
Okay.
graph is usually like this, that first it increases and at some point it becomes constant. Now here light is fully available, but still the reaction is becoming constant. Is it so?
Yes.
It means light is not a limiting factor here, because it
Okay.
is fully available. Means some other factor maybe. Now, what is our deduction? What that factor could be? This could be the chlorophyll, because only limited number of chlorophylls are present, or chloroplasts are present, and the light is absorbed by chloroplast. So if you have 100 units, let's say, if you have 100 units of chloroplast, each unit is absorbing 1 unit of light. So only 100 unit of lights can be absorbed.
Yes.
So if we are giving 200 units of light, the other 100 units cannot be used, because we have limited number of chlorophyll.
Okay.
So this is why we say light is not a limiting factor here, but some other maybe. Okay?
All right.
Okay, let's talk about temperature. Now temperature is a limiting factor. How it's a limiting factor? So when we increase the temperature, the reaction increases,
Yes.
but it's going to decrease after some time. Why do you think so?
Because uh because of transpiration.
Think
Like if
again. Temp is temperature. Think
oh
again. It's temperature.
okay.
So the graph would be first it increases and then it decreases.
Well,
What?
my initial thought was it would decrease because like I was thinking that if transpiration like started happening too much, then
Okay.
like
So stomata get closed.
yeah, so photosynthesis happens at like a lower rate then because there's like less diffusion happening or gaseous exchange happening.
Okay. What if I say think according to like some other chapter number 5. Go through chapter number 5 and tell me how the temperature affects it.
Okay,
Just
enzyme.
recall chapter number 5 and then
So
tell.
it'll be denatured.
It would be denatured.
Okay,
Okay?
okay, okay.
So we'll say it would be denatured. Okay. So why do some enzymes get denatured? How do you explain that?
We'll just talk about the enzymes that get denatured.
Yes, yes.
Okay.
How do enzymes get denatured, Shansi? I'm waiting for your answer.
Oh, okay. Basically, after they've reached the optimum temperature and they go like above that temperature, the active site get like the active site of the enzyme gets like deformed or denatured, and then the substrate can't fit into it. So it's not like able to be used.
Okay, correct. That because the temperature is so high that the intermolecular forces started to break, and they cannot like active the structure of active site is lost. The substrate cannot fit into the enzyme, and the reaction cannot take place.
Yes.
Okay. So our third factor is carbon dioxide. Carbon dioxide is similar to the light intensity. So it means carbon dioxide at some point the carbon dioxide cannot be the limiting factor.
Okay.